Hello, I'm James Duffin, and this channel is a series of talks about the control of breathing and some other topics of interest. In these talks, I present my own summary of current research, so be aware that they are my opinions and judge them accordingly. My aim is to provide a comprehension of how breathing is automatically generated and controlled. The series consists of related topics that eventually coalesce into a complete understanding, and this introduction briefly reviews them. So let us begin. The motor cortex up at the top has a direct control of the respiratory muscles. So one of the automatic systems of breathing control, we can actually take over voluntarily and do something about it. It's the only one. There are people, I'm told, that can control their heart rate, but not many of us. But in terms of respiration, the motor cortex will take over if we want. It competes, of course, with the rhythm generator that wants to keep us breathing. So that's why you can't hold your breath that long before you just have to breathe. Over in the frontal cortex, that is labeled because we laugh, we cry, and we are startled sometimes into saying things, and respiration is used for this too. The limbic system is a sort of relay of our general emotional system into the reticular activating system, which acts on respiration in terms of its rhythm and its transmission to the respiratory muscles. Coming down from there, we have the medulla rhythm generator, and very close by to that are the central chemoreceptors, which we'll be talking about. And these have axons, motor neurons nearby in the hypoglossal will send out respiratory signals to the tongue and certain muscles of the airway. So these are involved in things like sleep apnea. What could we do with something about that? But they also drive axons down the spinal cord where they will activate respiratory motor neurons, and then via the phrenic nerve, which goes all the way down here, we will activate the diaphragm. And later on, these same axons drive down into the intercostal motor neurons, which operate the muscles of the rib cage itself. We have feedback controls not only from the central chemoreceptors, which are looking at the local hydrogen ion concentration, but also from the carotid body, which is located here. It's a tiny body at the bifurcation of the carotid artery, and it sends signals back. It is sensitive to hydrogen ion concentration in the arterial blood going to the brain. So, as was said many years ago, it tastes the blood going to the brain to make sure that it is what we want. So it's an early warning system. Overall, we find that in terms of what the entire function of the respiratory system is, it is to protect the brain environment so that the brain functions well. We'll get into a little bit more of that in a moment. And so that is where we are overall in this picture. We can break this system up into various topics that we're going to look at. And I mentioned the topic of acid-base. That is of concern to us in our first topic because it's such a basic overarching theme that we need to know all about that before we can even start to think about other things. The other thing that is is overarching is the respiratory rhythm and the neural control system. And we can see that we have a neural controller in the medulla operating the spinal motor neurons, operating the respiratory muscles to produce pulmonary ventilation. Eventually, we'll get to how is respiratory muscle force translated into pulmonary ventilation, and that's topic 6 is pulmonary mechanics. There are local feedback systems within the spinal cord, and there are muscle receptors in the respiratory muscles that feed back. And so we have a local feedback here to control how the spinal respiratory motor neurons work. We have airway and lung receptors that will detect whether we're inhaling something that is noxious. It also feeds back information on how large the lungs and what volume they're getting to. That feeds back via the vagus nerve number 10 back into the central controller in the medulla. Pulmonary ventilation, of course, operates the lungs and controls the partial pressures of oxygen and CO2, and therefore hydrogen ion concentration by the Henderson-Hasselbalch equation. And these are detected by the peripheral chemoreceptors in the carotid bodies. They are sensitive to hydrogen ion concentration, but that sensitivity is modulated by the partial pressure of oxygen. And we'll look at carotid chemoreception as topic 4. Topic 3 will be central chemoreceptors. The central chemoreceptors in the medulla are sensitive to the local hydrogen ion concentration. And if we look at the whole system as a feedback control system, the complete chemoreflex control system, ventilation is controlled and we operate that ventilation to change gas exchange with the environment. That changes our partial pressure of oxygen in the arterial blood and the partial pressure of CO2 in the arterial blood. PCO2 affects brain blood flow and therefore affects the CO2 in the brain. And therefore, we translate what is happening to CO2 in the brain into hydrogen ion concentration, which is going to activate the central chemoreceptors. The same sort of thing is going on in the translation of CO2 into hydrogen ion concentration for the peripheral chemoreceptors, and PO2 acting as a modulator. Why is this a feedback control system, and we call it a negative feedback control system? Well, think of it this way. If I increase my ventilation for some reason or other, say I'm asleep and I roll over and I take a big sigh, that changes the gas exchange to lower this partial pressure of CO2. That also lowers the chemoreceptor input and therefore withdraws. In other words, it has a negative effect on ventilation drive. So it is a negative feedback control system as we go around that loop. Some of the other aspects that we're going to be talking about, I missed one, didn't I? I missed the fact that there are other drives to ventilation, such as exercise. And we are lucky that we have Dr. Daniel Kerr from Western, who's going to provide us with a talk on exercise in topic 7 on the control of breathing during exercise. We'll also challenge the system later on and see how it operates when we go up to altitude or we induce hypoxia one way or another. And we'll look at sleep. The emotions, as we said, run via the limbic system and reticular formation to affect the central controller of rhythm generation, as well as modulating the excitability of respiratory motor neurons in the spinal cord. It also acts as the relay system for any sensory stimuli like the startle reflex. If somebody fires a gun next to you, you jump, you immediately take an in breath, or if you jump into cold water, you suddenly want to inspire and breathe rapidly. So that effects go to the central rhythm generator there, which we're going to deal with in topic 5. Voluntary control, we said, yes, affects the respiratory motor neurons. There is a system of coughing and sneezing and hiccuping that also operates rather independently of these controls. And the cerebellum affects the respiratory motor neurons because as we bend over, we change the configuration of the rib cage and the operation of the phrenic nerve driving the diaphragm. So respiration motor neurons are affected by our posture. And of course, we have the rhythm generation itself. I've drawn these as separate pathways, and indeed they are. And so there are various things that occur if you have a spinal cord injury in an accident, for example. You may affect one or other of these pathways such that you may lose your voluntary control, but luckily your rhythm generation is still unaffected, and so breathing continues to keep you alive. You may also injure the rhythm transmission down to the respiratory motor neurons and yet be able to voluntarily control, and therefore you can stay alive with voluntary control. When you go to sleep, however, you're going to need a ventilator to keep you alive by breathing. Finally, the final topic will be a little talk on data acquisition, because presumably you're going to use instruments to measure things in either whole humans or various aspects of it, or in animal models. And we'll talk about some of the fundamentals of things that you should know. And that is the last topic, and it will be the one that does not require a written report. So here's an overall picture, rather like Seward Hung's, only in more detail. And it shows us where the rib cage muscles are and the lungs within this container, which we call the thoracic container. And you can see that our lungs must be filled via the airway. So we get our fresh air from the outside and it goes down the airway into the lungs. And this is a last in, first out system. Unlike things like birds and fishes, which have a flow through system, we have an inspire in through airways and then out through those same airways. Now, what does that mean? That means that the ventilation of the gas going in and then out of the airways does not do us any good as far as gas exchange goes, but it is simply a waste of our ventilation. And so we call that the anatomical dead space is called that dead space because it's wasted. I expect that you will have enough background, and if not, acquire it for understanding how the lungs oxygenate and remove CO2 from the blood via the alveoli here and across the alveolar membrane. And remember that the oxygen levels throughout the body from the lungs and the CO2 levels are driven entirely by the partial pressure gradient. Here's a little picture of the carotid body at the bifurcation of the carotid artery that he's shown us here. So one of the fundamental things that we're going to talk about is acid-base, and this is, we're going to use the modern quantitative acid-base chemistry as proposed by Peter Stewart. Peter Stewart is a Canadian, so be proud of your Canadian colleague here, now no longer alive, of course, but he published this in 1983. It's been rather controversial ever since, and so we still have papers like this that are quite new and say that we better start applying this quantitative acid-base physiology and replace the traditional bicarbonate-centered models, because most of what is taught in current medicine is simply the Henderson-Hasselbalch equation, which is the bicarbonate-centered model. So we're going to learn something that you may find quite different from what you've been taught. We'll provide you with this paper to read. I will send this to you almost immediately after this lecture, and I will send you a PowerPoint that is done by George here, and it's very good. And I have a phone call which I can get rid of. Sorry about that. And the next basic part that we need to learn to consider the overall system of respiratory neurophysiology, and specifically we're going to go into respiratory rhythm generation. We can go into that because it's still controversial. We still have arguments about how is respiratory rhythm generated, even after all these many years we've been studying it. Up at the top here, we have what's known as the RTN, the retrotrapezoid nucleus, which, as it turns out, is virtually what the central chemoreceptor is. These are the cells that sense hydrogen ion concentration and form the central chemoreceptor. Below that, we have what's known as the Betzinger complex, BOTS-C. No, it's not named after Dr. Botzinger who discovered it. It's named after an Australian winery. And there is a story behind that. Just below that, we have something that has come to be called the pre-Betzinger complex. Then we have a group of respiratory neurons that drive down the spinal cord, and not as shown here, they can go directly here, but they also drive the upper cervical inspiratory neurons, which Janusz Lipski and I investigated a few years ago in great detail. They're not called the upper cervical. We call them the UCINs, upper cervical inspiratory neurons. So getting back to these neurons here, which provide the drive to respiratory motor neurons in the spinal cord, we have what's known as the rostral ventral respiratory group. It's rostral because it's above the caudal ventral respiratory group, and it's ventral because it's close to the ventral surface of the medulla. And finally, down the phrenic motor neurons and the phrenic nuclei. Out coming from the phrenic nerve, which we often record, we can see that there is activity in this nerve which ramps up and then suddenly stops. Here's an integration of that. So this is what drives the respiratory muscles. And how is this rhythm generated so that it operates in this rather complex manner? We're going to find out. One of the things that people have been doing fairly recently, Matthias Deutschmann, my good colleague, just published this, where they have taken and volumetrically mapped the activity in a perfused brainstem perforation of rats. And that what that has done is they put down many, many microelectrodes to record throughout this volume of tissue in the medulla. And they've recorded the activity. And here is the time course of the activity. This is the phrenic nerve activity that they've recorded to show you when inspiration is occur. And this is the vagal nerve activity that is recorded as well. And I think if I do this, we will see the activity as it occurs in real time, and we will move the marker along here to view it. And you can see that where the activity begins during inspiration and at the end of inspiration, a lot of things light up. So that is a key feature of respiratory rhythm generator. It's easy enough to turn things on, but you now have to coordinate the turning off of respiration. So that's rather a fascinating look at what is happening, and it tells you all about the fact that there is a huge respiratory network that operates the respiratory rhythm generation that feeds down the spinal cord to the motor neurons there. The central chemoreceptors, we'll look at various aspects of that that have been studied in the retrotrapezoid nucleus, a couple from the Patrice Guillenet's lab. And they have not only been doing recording, they've been doing staining, they've been doing aspects whereby they can identify particular types of neurons and when they're active. There will be talk of photostimulation, of viral vectors, and something called DREADS, which does not refer to any hairstyle, but you'll find out what that means. And that will be a fairly complex topic because it'll introduce you to some of the aspects of how one does this kind of neurophysiology, which you will not be familiar with, and therefore cost you a fair bit of time to learn about them. But I think you'll find it fascinating. The peripheral chemoreceptors, here's the picture of them again. And if we look inside the peripheral chemoreceptors, we have glomeruli, which this is a picture of a glomeruli, which has glomus cells as its center. And the glomus cell is the heart of the system. It is what is detecting the hydrogen ion concentration in the blood going by in the blood vessels and releasing transmitters, as we see over here, that will affect the sensory nerve, which is the sinus nerve joining the glossopharyngeal nerve going up into the medulla. So that's a huge area of investigation. I'm going to tell you how I think it works. And here is a good review that we've taken pictures out of to show you here. And again, we'll be looking at all kinds of different aspects of neural transmission, what happens inside cells, modulation of calcium, ATP. These are subjects to which you may not have encountered before. So we'll be looking at those. Finally, we're going to put the whole system together, which is what I've been doing for many years with computer models. And here we have our feedback control system, ventilation, pulmonary gas exchange, altering the partial pressures of oxygen and CO2 in the arterial blood, leading to the various tissues. And of course, the brain, being the most important tissue, has its own central chemoreceptor, and it has early warning receptors in the peripheral chemoreceptors. So again, we notice that the brain is paramount here. We exist entirely to protect our brain and keep it functioning. Finally, pulmonary mechanics, where we will study the compliance of the lungs and how it expands as we control the interpleural pressure with our respiratory muscles, and what happens when we try and inspire and move air through the airways, which have a resistance to that movement, and the thoracic wall, the container of the lungs, the rib cage, that has a compliance too. And we'll find out how these operate together to make breathing easy for us. And we'll be able, once we've looked at this, to be able to find out what the work of breathing is. What is the energy cost of our breathing? Finally, we come to exercise, which Dr. Daniel Cure is going to talk about, but I've also been interested in for many years, and published this on the FAST exercise drive to breathe, which I presented at a conference. I think it was the American Physiological Society conference. And the big question about the fast exercise drive, which is the sudden increase in breathing as you start a rhythmic exercise. We call that the fast exercise drive because it occurs very quickly. There is a slow exercise drive to breathe, which Daniel will talk to you about. And we devised this apparatus whereby the experimenter can actually control the legs of the subject, and we can move the legs back and forth, or we can have the subject voluntarily move the legs back and forth. And this was able to answer a question that we had about where the fast exercise drive to breathe comes from. Does it come from the sensors in the leg muscles feeding back to control our ventilation? Or is it the fact that we are controlling our leg muscles voluntarily and some of that spills over to also affect our ventilation? That's called the central drive theory and the afferent drive theory. And so we did these experiments whereby you went along resting here, and then suddenly you were passively had your limbs move. And yes, indeed, ventilation suddenly increased. There's the slow increase as we keep going. Then we asked the subject to become active, and oh yes, suddenly we get another sudden increase, and we get a further slow increase again. When we add these all up, we find that they both contribute to a transition which would take us from rest to active exercise. And so that cleared up, at least in my mind, which drive was important. Both are. One of my former students, John Peever, who's now a professor at U of T, got involved in the control of sleep and wakefulness. And we'll be looking at that because when you go to sleep, there are aspects of respiration that are affected. I think we've all heard of sleep apnea, whereby the airway closes because the tongue falls back or the respiratory muscles in the airway are not active enough, and they occlude the airway so that you cannot breathe. And this causes CO2 to rise, and you are awakened and then start breathing again. But we also have different types of sleep, whether we have slow wave sleep, which is very deep sleep, or whether we have rapid eye movement sleep, which is dreaming sleep. And respiration is affected by both of these in quite different ways. So we'll find out about that too. Finally, we come to altitude, and my good friend Phil Ainsley in his laboratory has studied this extensively, taking subjects to altitude. So have we in our lab too, in the Fisher lab, and I have been involved in altitude experiments by other labs as well. And you see that as you acclimatize, as you travel to altitude, there is a fairly rapid increase in the ventilation, which then falls away in the first 10 minutes. And then we have something called hypoxic ventilatory decline, HVD. This is the so-called the hypoxic ventilatory response. But then as you continue over several days, there is a slow acclimatization. How does that occur? are the peripheral chemoreceptors involved? What about acid-base changes? Because we're changing the CO2 by hyperventilating at first. There is a lot to understand in the complexity of these responses. And the cerebrovasculature, that also responds. And the question is, if you change the cerebral blood flow, do you change the hydrogen ion concentration and the CO2 in the central chemoreceptor area? And I'm going to give you a hint that maybe we do, maybe we don't. Let's find out eventually. And finally, we get to data acquisition. And here is one of Dr. Kerr's experiments, whereby we can look at various things and record them. And we're recording the partial pressure of oxygen and the partial pressure of CO2. And we are changing them to find out what happens to blood pressure, what happens to tidal volume, breathing, and what happens to the muscle sympathetic nerve activity. This gives us an output of what the sympathetic nerve is doing to control the peripheral circulation. So here we're controlling the PO2 to be constant at a high level, and here we're putting it at a low, a hypoxic level, 150 and 50 PO2s. And here we use a technique that I invented several years ago called modified rebreathing. The original rebreathing technique was invented by David Reed, an Australian, but I have modified it in a few ways. And here we see that we are going along at our resting CO2. We begin to rebreathe, we equilibrate, and then the CO2 rises slowly over the next 4 or 5 minutes. Same thing going on here. And you can see that indeed we get a rise in blood pressure. We go along with ventilation, nothing much happens until we reach kind of threshold, and then we increase our ventilation. This threshold seems to occur earlier if we are hypoxic. And we notice that the activity in the muscle sympathetic nerve activity increases here as the CO2 gets higher, even more so when you are hypoxic. And the interesting thing is that the threshold for this increase in ventilation is also at the same partial pressure of CO2 as we find for the increase in muscle sympathetic nerve activity. So that's an interesting story as to why that should be so.